Good day. I am Dr. Priyanka from Department of Physiology, Sree Balaji Medical College and Hospital, affiliated to Bharat Institute of Higher Education and Research. Today, the topic of my discussion is apoptosis. At the end of the session, the student will be able to define what is apoptosis, differentiate between necrosis and apoptosis, describe the physiological basis of apoptosis, along with the morphological changes happening in apoptosis, and the molecular mechanisms that takes place in the apoptosis. Let's go for the class. Now let's see a brief history about apoptosis. It was first Carl Wojet who first derived the principle of apoptosis in 1842, after which Walter Fleming described about the process of programmed cell death, and it was first John Ross Kerr who first introduced the term apoptosis in his publication in 1972, followed by Sidney Berner, Horwitz, John E. Sulzenson described the Nobel Prize in Medicine, received a Nobel Prize in Medicine for their pioneering research on apoptosis in 2002. Apoptosis or programmed cell death, it is defined as a highly regulated active cell death characterized by cell shrinkage and nuclear condensation. It is pre-programmed and there is a number of cascade events taking place which leads to morphological changes, and it is just opposite to necrosis. Let's see the differences between apoptosis and necrosis. Apoptosis is physiological or it may be pathological and it is specific. Necrosis is usually physiological or it is usually traumatic or accidental. Active process takes place in case of apoptosis, and it is totally energy dependent and new proteins are always synthesized. Whereas in case of necrosis, it is a passive process, it is energy independent and there is no protein synthesis observed. There is presence of membrane blebbing in apoptosis, whereas there is disruption seen in case of necrosis. The cytoplasm shrinks, there is cellular fragmentation, and there is membrane bound vesicle formed, which is called as apoptotic bodies, are seen in case of apoptosis. In case of necrosis, there is no such vesicles formed, but the cytoplasm swells and the cell lyses. Therefore, in case of apoptosis, non-random lengthening of the fragmentation of DNA is observed, which shows a ladder pattern in case of gel electrophoresis. Whereas in necrosis, there is random digestion of the DNA, which leads to a smear pattern in case of gel electrophoresis. Usually, individual cells are affected in case of apoptosis and there is no inflammatory response seen. Whereas in necrosis, there is significant inflammatory response seen and it is usually a group of contiguous cells getting affected. Apoptosis is usually by means of a physiological stimuli, which can be due to lack of growth factors, altered hormonal environment, etc. Necrosis is usually due to non-physiological stimuli such as hypoxia, hyperthermia, viral infections, etc. Why should a cell actually undergo self-destruction? Programmed cell death is essential for proper development. Examples: a tadpole, tail needs to be resorbed at the time of metamorphosis into an adult frog. Formation of fingers and toes during the fetal development. Slowing of the inner lining of the endometrium at the start of the menstruation. Formation of proper synaptic connections between the neurons. Elimination of T cells. Otherwise, there is huge amount of autoimmune attack going to be happening in the body. Thus, programmed cell death is essential for proper development of a body. Apart from that, the programmed death is also needed to destroy cells that represent a threat to the integrity of an organism. Example, if the cytotoxic T cells are usually appointed to kill the virally infected cells, thus it induces apoptosis. Removal of the DNA damaged cells for proper embryonic development. Radiation, chemicals used in cancer therapy induces apoptosis. Effector cell removal to prevent autoimmunity. Usually, apoptosis, if not noted, then it leads to a number of disorders, such as if there is excess apoptosis, it leads to AIDS, stroke, Alzheimer's disease, Huntington's disease, Parkinson's, amyotrophic lateral sclerosis, traumatic brain injury, spinal cord injuries. Diseases linked with inadequate apoptosis, examples are cancer, SLE, systemic lupus erythematosus, multiple sclerosis, diabetes mellitus, and rheumatoid arthritis. Thus, apoptosis is need to be learned and concentrated more. So let's go for the class. Apoptosis. What is apoptosis? Apoptosis is simply called as programmed cell death. The major morphological changes that we can observe in an apoptotic cell or apoptoized cell is shell shrinkage, chromatin condensation, formation of cytoplasmic blebs or apoptotic bodies, and these apoptotic bodies are phagocytized by the macrophages later on. So these are the major histopathological slides which you can observe the apoptosis taking place in different systems of your body. The first one is the apoptotic cell in the liver injury. The normal cell is available on besides and the apoptotic cell size. And the second one shows the chromatin condensation below the nuclear membrane. And the third one is an impression of apoptotic bodies that is seen in the pancreatic tissue. And soon after the apoptosis, the cell size starts to shrink and we can see dense eosinophilic cytoplasm and the chromatin starts to condense to form clumps. Depending upon the etiological factors, apoptosis can be initiated by means of intrinsic or mitochondrial pathway of mechanism, extrinsic receptor initiated pathway mechanism, or it can opt perforin gran enzyme pathway mechanism. Going for the intrinsic pathway of mechanism, usually the cytochrome C is present inside the mitochondria, which is normally not seen in the cytoplasm. When there is any disturbance in the cell, such as it can be due to radiation or it can be a protein misfolding which happens in case of mutation, or if there is stress protein stressing the mitochondria, there is a lot of changes happening in the mitochondria which will cause leaking of the cytochrome C into the cytoplasm, which will actually lead to apoptosis. Let's see that. So if there is involvement of radiation or protein misfolding happening, the integrity of the mitochondrial cell membrane gets disturbed, which will cause the activation of BCL2 and BCLX, which in turn will cause activation of the leaking of the cytochrome C, and it also causes release of stress proteins called as BH3 only proteins. Those three proteins are BIM, BAD, and BID. BIM, BAD, and BID. These three BH3 proteins will prevent the function of BCL2 and BCLX, which are anti-apoptotic in nature. Plus, they also activate the two pro-apoptotic factors called as BAX and BAC, which allows the cell membrane of the mitochondria to ooze cytochrome C into the cytoplasm. Soon after, the release of cytochrome C into the cytoplasm will cause the cytochrome C to go bind with the protein called as apoptosis activating protein factor number 1. It is called as APAF1 shortly. After binding of this apoptosis activating factor 1, the cytochrome C will cause the activation of caspase 9. Caspase 9 are initiator molecules which in turn will activate the executioner caspases number 6 and 3, and thus the intrinsic mechanism of apoptosis starts to take place. Once again I repeat, in case of intrinsic mechanism of apoptosis, the cytochrome C is safely sealed inside the mitochondria in case of normal status. During pathological status, it can be due to radiation or mutation or release of stress proteins will cause the release of stress proteins called as BIM, BAD, and BID, which will block the function of BCL2 and BCLX, which actually protects the mitochondria. So when these two proteins are blocked, it goes and allows the release of cytochrome C into the cytoplasm from the mitochondria. And the released cytochrome C will go and bind with the apoptosis activating factor number 1 to form apoptosome. This apoptosome in turn will cause activation of initiator caspase number 9. After the activation of initiator caspase number 9, it will tend to initiate the executioner caspases number 3 and 6, and the apoptosis will start to take place. Next comes the extrinsic pathway of apoptosis, which involves cytotoxic CD8 T cells, cytotoxic or CD8 T cells. So, cytotoxic CD8 T cells usually will attack the infected or tumor or damaged cells. The T cells will tend to express fast ligand on its receptor, whereas the infected or damaged cells will tend to express fast receptor on its surface area. Once the fast ligand of the T cell and the fast receptor of the tumor cell starts to interact, there is a number of apoptotic signals getting released which will cause the activation of extrinsic pathway, or it can opt gran enzyme perforin pathway of apoptosis. Let's see one by one. The extrinsic mechanism of apoptosis takes place as follows. The fast ligand of the T cell and the fast receptor of the infected tumor cell, once they start to bind, they activate a death domain called as FADD, fast associated death domain. Activation of this FAD will pull caspase 8 to its surface, and binding of these 4 molecules, fast ligand, the fast receptor, the FAD, death domain, and caspase 8, all 4 together will initiate the activation of caspase 8. Once caspase 8 gets initiated, they tend to activate the neighboring caspase 8. Once they get activated, the caspase initiator 8 number will tend to go activate number 3 and 6 executioner caspases, and the extrinsic mechanism of apoptosis starts to take place. The last way of apoptosis is release of GRAN enzymes. The release of GRAN enzymes involves the T cells once again. The T cell will release a substance called as perforins, and these perforins will form pores on the transmembrane of the cells, through which the GRAN enzyme is another substance produced by the T cell will tend to enter into the infected cells. And after it is entering into the infected cells, there is once again activation of executor caspases 3 and 6 directly, and then it causes DNA cleavage and leading to apoptosis. Once the apoptosis gets executed, the cell starts to bleb and gets fragmented into small pieces, and these pieces will be coated by a special substance called as phosphatidyl serine, or it can also be coated by immunoglobulin G complement protein, and then it is phagocytosed by the macrophages. So once the apoptosis takes place and caspase 3 and 6 disintegrates the DNA, and it also damages the cytoskeletal protein, leading to cell shrinkage, nuclear fragmentation, blebbing, the apoptotic bodies, and finally the phagocytosis of the apoptoized substances. Coming to the end of the session, I believe I made myself clear about this topic. Every second, almost 1 lakh cells in the human body die by apoptosis, while a similar amount are generated by mitosis. Thus, apoptosis is very important, and it has its implications in cardiac remodeling, pancreatic cancer pathology, and apoptosis acts like a stone sculpture which tries to reveal new shapes every day in the body systems. Thank you.